Okay, guys, we are live on Facebook. If everybody can just stand by, I'm just trying to get us live on live on YouTube. I'm trying to get us live on Facebook. So if you can bear with me 1 minute, we'll join you in just a second. Okay, good evening, everybody. Sorry about the delay. Just had to do a little bit of work, multitasking here. Welcome to Falkor Live. My name is Deborah Smith. I'm a marine technician here on the RV Falkor. Falkor is owned and operated by Schmidt Ocean Institute. We thank you for joining us this evening. I am joined with a number of scientists from this expedition who are going to explain to you how they use the wet lab on Falkor. So Falkor is unique, not unique to any other research vessel, but unique that we have a number of different places on the ship for scientists to be able to do some work. So we have our control room, with which you see me in now, which we've talked about on previous live feeds. This is where the ROV is controlled from, where we do mapping, which we're currently doing right now. The weather is a bit rough this evening, so I ask you to bear with our video feeds. You will actually probably see the swaying of the ship. The ship is eminently about to turn around and head into the seas, so you never know what you might see fall over. But thank you very much for joining us. I ask you hold to questions so a little bit later in our show, our scientists are going to first introduce themselves, explain a little bit about the instrumentation and the equipment that you find in the wet lab, and then they're actually going to show you a sample that they collected earlier today and some of the processes they go through, as well as some of the other types of sampling and processing that happen in our wet lab. So I'm going to send it over to the guys in the wet lab and have them introduce themselves. Go ahead, guys.
Hi everyone, hi to our friends and family at home. My name is Georgia Nesta and I'm a PhD student at Curtin University. I'm also a student of opportunity on the Falkor. So what I do on the ship is conduct environmental DNA analysis, which I'm sure you've probably heard about through our live streams. I'll go over it a bit more and I'll show you it in detail. I'll over to David.
So my name's David Yushkevich. I'm a PhD candidate at Curtin University and also a student of opportunity with the Schmidt Ocean Institute. So my job has been to support the WA Museum in the biological sampling process from the moment the samples come up from the ROV through to the processing in this wet lab. So we'll start today by actually giving you a bit of a tour around the excellent wet lab facility on board the RV Falkor. So we'll start here. So like in most labs, we've got an ice fridge. So provides us ice to cool the samples as they come up and any other cooling needs. We've got a light microscope. A lot of the bench spaces have got bolts to kind of secure everything in the listing and rolling that occurs during our travels. So we've got an array of refrigerators and freezers. Here we've got a fridge that goes down to 4 degrees where we're storing most of our samples. We're also cooling a lot of the water as the samples are coming up from 1 to 4 degrees, so we want to stabilize the samples when they come up. We also have a freezer that goes down to -30. Brief. So here we store some of our ethanol samples and also we cool the buckets. We also have a -80 freezer, which is where our final ethanol storage samples will go, or if any samples cannot be preserved, then we'll end up in here until we disembark. Just take you around. So we've got our waste disposal around here, so we kind of make sure we minimize waste. We're quite careful. We make sure that everything's always put in its place, so to avoid slips, trips and hazards when we're all working together, because when the samples come up, we all need to maneuver around each other. There's a lot of us in this lab working on different projects, from Lisa Nerida working on the mollusks to Andrew Hosey working on the crustaceans, to Glenn Moore working with the fish. So we all have to work as a team and ensure that everything's secured. Like any lab, of course, we've got fire prevention systems in case of a fire. Some fire extinguishers here. If you move over through here, so we've got sinks like in most labs. The facility is really great, like a lot of the facilities you find in universities and institutions. The wet lab is really great, actually, for the needs that we have. So we've got a lot of non-slip matting. It's placed down on these awesome bench spaces where we can conduct our work, because, of course, when the ship is listing and rolling, there's a lot of movement. We're dealing with chemicals, dealing with specimens, so we just need to ensure that we can stabilize everything, work around each other conveniently. So plenty of cupboards, plenty of storage. So arraignment, there's storage everywhere, which is fantastic. We can put our gloves, we can put all our different equipment, make sure it's packed away at the end of the day. Coming across here, we've got special designated cabinets, as in most labs. So this has been fantastic, so we don't have to store them somewhere different on the ship. So we've got an area for our flammables, so this is where we keep our ethanol in our situation, which is where a lot of samples are being preserved. And then we've got a fume cabinet, which is excellent to actually have this in the wet lab. So we're dealing, for some specimens that are gelatinous and soft-bodied, we preserve these in formalin. And this is really important for IDing species, to make sure that the sample is actually kept its shape. And formalin, very dangerous chemical, and of course we don't want to be breathing any of those fumes in, so a lot of that stuff gets done in this fume hood where it's got an extractor, and any fumes will go through the system and not into the air around us in the lab. So we've got lots of bench space, which is fantastic. Lots of eye bolts where we can secure stuff like the dissecting microscope, our camera equipment. Of course, most importantly, when working around chemicals, we've got spill response kits. So there's a cabinet here that has our spill response kits, the different types of chemicals that we're using, just in case we do have a spill, because we are working on a boat that's moving around and that can happen. So I think we'll go
Through your sample?
The sample, yep.
Perfect.
So today we sampled in Clotes Canyon. It was a short dive, but we're still very lucky to find some really interesting stuff. So I'll just go with you through the process. So once the samples have come from the ROV, the first job is to actually put them in trays and put them straight into the refrigerator. And this makes sure that the samples are cooled and stabilized in the same water temperature as what they came up with. So this is usually about 1.4 to 4 degrees. So the next step is we then assign each specimen with a unique WA museum number. So most museums will have an allocation of numbers. So what these labels would look like. So this is a very important label. It's also used for provenance of the actual sample itself. So in this case, what we would write is we would write the type of specimen. So what I'll do is I'll just gently lift this up, and we have to be really gentle with a lot of the specimens that we collect. They are very fragile, and some of these specimens could be holotypes. So the first species is used for the first specimen used for the identification of that species. This one here, we've got a beautiful orange sea star. So we would firstly label what it is. So it's just a general labeling, because of course you need trained professionals within taxonomy of that group to actually identify to a species. In this sense, we've just called it an asteroid, which is the group of sea stars. Secondly, we'll then put the ID number of when the sample was collected and the actual dive. Then we'd put the date, and then an allocated number is given to this sample. So first thing, we'd obviously have some gloves on, so most importantly to this. So the first step in all of this is the sample will get put onto a tray with ice. So we've prepared this earlier, and we ensure this that then the sample is sitting on an ice tray and it is constantly cooled, right temperature. So the sample itself will first undergo a photo shoot. And this is really important because after the sample is preserved in nature, it won't look the same as it was when it was first collected. So the beauty of collecting stuff with the ROV is we have some beautiful 4K in situ photos. In situ meaning collected at the time, you know, where they're living in its habitat. But of course, we still want to photograph the specimen itself. So we use an array of flashes, and this really does become a photo shoot for the sample itself. And this is very important, as specific taxonomists that work on different taxa need different characters. They use different morphological characters to identify the specimen to a species level. So,
just for reference as well, we collected this starfish at 2,000 meters. Okay.
That's fantastic. It's amazing some of the stuff we've been collecting. Really beautiful specimens. We get Georgia, so the most important thing is, of course, we start every photo with its actual allocated number. We would put that into the first group of photos. And I'll just check that the flashes are on. So every specimen needs to be photographed differently. Sometimes you need the mouth, the feet, the different characters. So with sea stars, with any specimen, firstly, the most important photos are the actual whole specimen itself, both sides up. And with a sea star, some of the important characters would be the actual mouth itself, and also the end or the tips of the arms. So that's because there are different plates, and the way the different plates are fused together can matter for morphological identification. In this case, I'll just zoom into the mouth and take a photo of that.
David, can I interrupt you real quick? Sorry.
Yep.
Can you just explain to people at home why those are really important, why all those different features are important for you guys to capture in these really high-resolution close-up imagery that you might not get from the ROV, which can look at something from really far away?
It's mainly just for morphological identification, isn't it, David?
Yeah, so in this modern day, we use a lot of integrated taxonomy. So that means we couple morphological characters along with molecular work. And in situ photos also provide a lot of information. We get a lot of information from the ROV, the depth, the GPS locations, but of course the morphological characters underlie the fundamental historical ways of us IDing species, and so that's where a lot of work begins, based on the morphology.
Can you explain what morphology is to the kids at home?
Just like its characteristics. So say if this starfish had lots of little bumps or lots of little teeth, and then another starfish we had like bumps instead, and it was completely barren, we would identify those as different species.
Yeah.
So it's just like how me and David look completely different.
So what's really cool about these deep sea stars is they're a bit unique because they actually have a plate on the very end of the arms, so that's quite unique about these ones. So that's one of the photos that we're capturing.
Great, thank you so much. I'll let you guys carry on. Sorry for the interruption.
No, that's fine. As you can see, with the boat listing and rolling, it can be quite difficult to photograph. And of course we have this big black screen here to avoid any reflections. So because we are taking the specimens in water, when the flashes go off, if our hands are in the way of this black sheet, the reflection comes up in the photograph. So that's why it's quite important to isolate photograph in. Just try and get this photo. This can be quite tricky sometimes, but I feel like we have mastered it now. So here, just show everyone at home. Here you've got this beautiful, so you see a lot of this information can be very useful. There's the mouth itself there. A lot of this will come up in the preserved specimen, but it helps the taxonomist with the initial.
When it's fresh still. Yeah.
As much documentation as we can do before it gets preserved, it all helps along the taxonomic journey of IDing a species. So, next step that we will do after giving our specimen a photo shoot and recording all this is the latest thing that we do is we use molecular work. So what we'll do is we'll subsample a bit of the tissue. So we'll take a little bit of the organism and preserve it in 100% ethanol. And different specimens need to be sampled differently. So some of the sea cucumbers that you've seen us bring up, we would sample the tube feet with the sea star. We try and get in between the grooves to sample some of the feet through there.
Is that mainly just because get to those parts on?
It's because of the amount of DNA.
Right.
So some of these harbour more DNA than other places. So if we were just to chop off a bit of the arm, you have more plates there, so you wouldn't get as much DNA, and that's quite important. So Glenn, for example, who works on the fish, he will take a snippet of flesh out of the fish itself under the scales. So it's always important to reduce contamination with DNA, as you'll probably explain later. So what we will do is we have this container of vials, and the number that the specimen has been given, we'll print a label, circle label with that number, and we'll place that number onto the tube. This can also be done by pen. It's not a big issue, but it just keeps things a bit more organised and a bit more systematic. What we would do is we'd grab the specimen, and of course we'd always handle it with care. Is we take a bit of the tissue, because we're putting in 100% ethanol and we want to preserve the tissue sample best, we don't want to dilute ethanol any less to what it already is. So we'll dry the sample, a bit of Kimtech sterile wipes just to remove the excess water. These absorb a lot of that excess water really well. Yep, and Georgia will just open up the vial. And then we'll just place that into the vial, and that will be stored. And of course we'd clean our forceps between all samples to avoid contamination. And then this container would then be put into the freezer at -30. So then after our photo shoot, after we've subsampled, we'll then take the sample and we'll preserve it. So different specimens get preserved differently. Because this is a hard-bodied sea star, it's not gelatinous, we wouldn't put it in formalin, but we'd put it in 75% ethanol. So I can show you how that step goes. So we go to our flammables cabinet where we store our ethanol. Being really careful. Go over to the sink. Get Georgia, if you can unscrew that. So, because we need to minimise storage with the way everything gets transported to the museum, we've either got containers, dependent on how soft the sample is. We're basically putting stuff into barrels, and so we want to ensure that the sample gets transported the best way, but of course efficiently to reduce space. The sea star can be placed in plastic bags until we transport into the museum, where they'll be put into proper containers. But for the purposes of transport and reducing space, a bag's fine. So what we would do is we'd get the bag, we'd put its allocated label into the bag, grab the sea star, the specimen itself, place it into the bag, and then Georgia will assist me then to flood the specimen with 75% ethanol.
God, I thought you wanted me to pour it.
No. Of course, all we're doing is we're just trying to cover the specimen. I'm going to try and get as much air out as we can in the bag. As if it was in a plastic container, some of the holothurians, the sea cucumbers, because they're very gelatinous, we would put them into a tub or a little container. So we'll get as much air out as we can. There's our sea star curved. And then we'll double bag that just to ensure leakage. And then that sample is ready to get put away. Transport. We'll just leave this in the sink for now. So then after we've dealt with all our specimens, we'll then pack them away into barrels and lock them into the corresponding containers to make sure no ethanol could escape, cause a hazard. And I think I'll leave it to Georgia now.
Awesome.
Talk about eDNA.
Thank you, David.
Georgia, real quick before you start, let me just show the people at home. If I could have you narrate a little bit, I'm going to have Casey keep the camera on you, but if you look up a little bit in the corner at the TV, I want you to narrate a little bit about what's going on, so you can see you.
Sure.
It's gonna re-loop.
So in this photo, this is what it would actually look like if I was sampling. It's a lot more stressful than what I'm about to show you. So we take our samples off of the ROV. So I have water samples. I'll explain a little bit about eDNA first. So eDNA is environmental DNA. So every living organism has DNA. For example, they shed DNA into the environment through mucus, through feces, through their tissues. We can take a water sample, and then from this water sample, we can detect what species are living in that environment at that time. So with what I do on the Falkor is we take Niskin bottles, so they're just big plastic bottles really, and we just program them to open up at a certain depth. It collects 12 litres of water, and there's 4 of them. My math is terrible. They all open up at whatever depths we program them to. When they come onto the surface, we have to be quite quick to get that water out and then take it down to the lab to filter. Environmental DNA is quite finicky in the sense that it's very fragile and we need to act quick because it can degrade quite quickly. As hot as the water gets, it'll degrade quicker.
Georgia, can you talk a little bit about, before you get into the super details of eDNA, can you talk about sort of the general picture of eDNA? What are you trying to capture by collecting this water?
Yeah.
A little bit like what is the concept of eDNA and the idea of taking water at different depths?
Yeah, so in this study, we're trying to capture all the biodiversity of the Ningaloo Canyons. With the ROV, we have the video imagery, and then we collect samples and physically bring them up to the surface. But there could be lots of things that we're missing visually and lots of things that we're not collecting. So environmental DNA, we can detect species that we might not have seen through the ROV or the species that we might not have brought up. So that's what the good thing about eDNA is that you can detect species that you might not have visually caught otherwise. That's what we're really trying to do here with that.
Awesome. So the video has just done its loop. Everybody at home got to see a little bit about what happens in a wet lab, all the different aspects, as well as this last bit, which I'm going to come back over to Georgia. And here you go. Here's Georgia. So you can talk a little bit more about what's happening in those imagery with that water.
Yeah, perfect. I'll show you like a quick little demo as well. What we would do normally is the ROV comes up, we empty the water into big buckets. My lovely assistant Casey helps me carry them down the stairs into the lab. Or if we're doing a CTD, so a CTD is just like a big machine. We would love to show you, but it's a bit rolly today and it's a bit dangerous out on the deck. It's a big machine that has lots of Niskin bottles, so lots of those 12-litre bottles I was telling you about. And we program that to open at different depths. The reason I want to look at different depths is because the biodiversity, all the animals living in an ecosystem, changes with depth. On the surface, we have lots of fish because we have lots of sunlight, lots of food availability, whereas in the deep sea, there's not maybe not as much, but completely different biodiversity. So we just want to capture that throughout the water column. So I would have my buckets of water on the floor. I don't have that, obviously, for you, sorry. So it's going to be boring old tap water. We would fill up a sample, and then I would want to run it through my filter system. So this is a filter pump. It's a peristaltic pump. So what it is, this is the actual machine, and this is just the cup that I filter it through. So this, we would place a filter paper onto here, and I'd filter all the water through. So on this filter paper, all the DNA will stay here. So at the end, I end up with just a tiny little piece of paper that I'll put away in a sample bag and put in the freezer. So like David's stuff, we store eDNA at -80. It's really good for preservation. It's quite fragile, as I mentioned, eDNA, so we try to keep it as cold as we can for as long as we can until we extract all the DNA and try and figure out what lived in that environment at that time using lab techniques. So, and this is a real thing. Pour the water in here, as exciting as David's, I'm sorry, and then the noise that haunts my nightmare, filtering.
The results are very amazing, though.
Yeah, my lab work can be sometimes not as exciting, but yeah, my results are often pretty amazing. The great thing about eDNA, and the reason I love eDNA so much, is that it's a very non-invasive method, and you do get to capture a wide range of biodiversity by targeting different DNA and different species. Yeah, we would filter that through. I would take the filter paper, put it in a little bag that I have everything labeled anally, and then pop it in the freezer at -80.
Georgia, can you show us the inside of the -80?
I can diddly do, but it's down the stairs, Deb.
I know, but you can use this one.
You can pretend.
Because I just want people to see how cold that looks when you open up. Wait for Casey.
So cold.
It's like snowing in Europe.
You honestly, if you touch the metal, like my hands burn. It's horrible.
It's incredibly
then it's
cold.
just touching my back, which is delightful.
Thanks.
It's the -80.
So for everybody at home, that's
fine,
-80 degrees Celsius.
Celsius, yes. The correct usually.
Georgia, there's a question from our online viewers. Is that 45 micron filter? Do you know what size filter?
A DNA person. I use 0.45s. I feel like this is probably someone in our lab trying to trick me up. See, what they're talking about there in the micron filter is the smaller the filter size, the more likelihood you have of capturing DNA in that environment. So if you use a filter size with huge pores, the water's just going to flush right through, and you're probably going to miss some of the small cryptic stuff. I like to use smaller filter papers, so I use 0.45s for the ROV stuff for a comparison, and then for the Niskin stuff, because I filter a massive amount of water, I use 0.8s. That answers your question.
Awesome. Okay, so I'm going to just jump in here real quick and ask a few questions. But while I'm asking them a few questions, if those of you at home would like to ask David or Georgia a question, please feel free to write in, and I will relay those questions, and we'll see if we can get them answered. First of all, could one of you explain why it's really important to have -75? They were just actually, this is one of my jobs, when we get lots of alarms, I'm not sure if you've been hearing it at home, but there are lots of beeping in this wet lab. The freezer, every time you open that -80 freezer and it goes up to -75, it warns you and tells you that it's not at temperature. Same thing with that lovely fume hood. Every time the flow is very low, it warns them. So we, in the Marine Tech department, annoy the scientists quite a bit with all of these alarms that go off. But
just ignore them, Deb.
Yeah, that's good.
No, I'm kidding.
So why is it important to have a wet lab on a ship?
Well, I don't think I could physically do anything if we didn't have a wet lab. Having a wet lab means that we can actually process our samples on board. I think to date I've filtered 12 to date on this ship, 1,200 litres of water. If I had to freeze that and take it back to Perth, filter, I'd be in a world of pain.
It's also it's a lab that's been created for a space where we can work with chemicals, we can work with wet samples. So most of the time you'll have a dry lab and a wet lab. So dry labs where we would conduct a lot of our laptop work, analysis work, monitoring of the drops of the CTD.
Data entry, all of that exciting stuff.
And then the wet lab it's got all the facilities, it's got the fume hoods, it's got the safety equipment to deal with any spills. Everything's kind of facilitated. So we've got taps, we've got freshwater flows, we've got seawater that's been filtered out that we can then top up our samples if we're running out of water or if we need to dilute things. It's a space used for a lot of biological sampling processing for George's work.
Yeah, it just means as well that we can start our work straight away, process it straight away, and then make sure our samples are the safest that they can possibly be and as preserved as they can be. We work when the ROV comes up and the CTD comes up, we work to get those samples in the freezer. And
it is called, it's not called a wet lab because we get drenched in here, but there is maybe not some of us, but there is water going here and there. There's water in the trays, we have chemicals, so it's important there's proper drainage in the labs, there's proper ways to mitigate spills and to deal with these things.
Awesome. Thanks so much for those answers, guys. We do have some questions coming in from online. Georgia, does the DNA sequencing happen in this lab as well?
No, it doesn't.
That'd be cool, though.
Yeah, I wouldn't sleep ever.
Get a PCR on board.
Yeah, so the DNA sequencing, I'll explain it a little bit. So like I mentioned, I have those filter papers. We take that back to the lab, and then this person who's asking about sequencing, what that means is we extract all the DNA from that sample and sequence it and figure out what species were in there. So the sequencer is the actual machine that tells me, it just gives me a bunch of A's, C's, G's, and T's, a bunch of letters that make no sense, and then I have to do a lot of programming on the computer to figure them out. But unfortunately on board, well, not unfortunately, because I wouldn't be able to sleep, but we don't have a sequencer on board. The sequencer is back at Curtin. These samples will stay frozen and preserved with me, and then I'll bring them back to Curtin University and process them there.
Great.
We do have a, sorry Deb, little mini sequencer that we sometimes take out, but it can only do a certain amount of samples. But the amount I'm doing now, it's not.
That's, if I can add to that, and that's why it's so important while we take the tissue samples. This is why we sample our tissue samples from the specimen itself. So that will then undergo sequencing as well using special primers. So primers are like bookmarks, which target a different region of the different organisms. And then this information ends up going on GenBank, which provides a reference database for any scientist to use. And Georgia will, of course, use a lot of this information in her eDNA studies.
Yeah, that's an awesome thing about it, is that all the samples that the guys are getting here in the lab, they're going to sequence. Then I'll have a reference library to compare my results to as well. Yeah.
Great. So there's a question from James. James has asked two questions. So the first question is to David. When you took your photo, I did not see a scale. How do you measure features from the photo without a ruler? So do you normally have some sort of measurement scale when you're taking pictures of all of these samples, or do you do some sort of measurement of your samples?
So James, thanks for that excellent question. So normally we would have a scale in the actual vessel itself that we're photographing, but because in this case we're physically just trying to, teaching purposes and to present to you guys what's going on, we didn't want to kind of draw out that long process. The other thing is a lot of the photos that are taken, even if you didn't have a scale bar, and these are some of the things that I've dealt with, you physically have the specimen itself, can still correspond the photography to the specimen itself, and that's what's so beautiful about having museums like the West Australian Museum, where they deposit these important specimens. And as scientists, we can contact people like Nerida Wilson, Lisa Kirkendale, all these awesome scientists that are on board who work at the museum, and we can request to come and visually go to the WA Museum and look at the specimens that they harbour within their awesome facility. So we can still actually obtain measurements even if the scale bar isn't in there, but we would normally put a ruler in the actual vessel itself. I hope that answers your question, James.
Great. Okay, so the second question, there's two more questions from James, but first I'm going to address another question. So the question is about, do fish die when they come up from a very high pressure to the surface? And unfortunately, the answer to that is yes, they usually do. All of these specimens don't make it back to the museum alive. That's not necessarily the purpose of that. It's to preserve and to learn more about these species. The scientists only take what they need, only take the number of samples that they need to get an idea of what is out here in that biodiversity, but they're not taking every one that they come across. And so they've been very good about being selective about what they choose, because we know that those samples are probably not going to come back.
Yeah, and a lot of the samples that we have found are new records or stuff that we've never seen here before, so we do need to collect them and collect their DNA, and then it's also just great for the museum to have a record. So we normally collect just one or two.
Yeah, the basis of a lot of ecology and scientific work based on organisms starts with the actual organism itself, and we can't carry out any work and any development of how things are changing with climate change or a lot of other work without actually knowing what species they're working on. And this is where the fundamental science begins, with taxonomy, with the collections, and trying to understand what occurs where. And
once we collect that sample, I'll have a record for my eDNA, and then we can do more non-invasive surveying methods as well, which is perfect.
Yeah, and it just stems from other people coming back here and maybe wanting to know more and targeting a specific organism. So this work is really important, and unfortunately sometimes we do have to sacrifice organisms for the greater good.
Love them though.
We do. It's always hard to preserve them, but yeah, it's something we do have to do for future science.
Okay, so there's a few more questions. A bunch of questions about eDNA. Georgia wins the question award tonight. One question is, how long does it take to process an eDNA sample once you get back?
Oh, too long.
You've been gone for a couple of years now, right?
Yeah, that's the issue with eDNA. It's a slow process, but it's a rewarding process. So it does take quite a while to do all the lab work, mainly because you have to do everything quite carefully to avoid contamination. So between samples, I'll be changing gloves, tweezers, benches, everything has to be bleached and ethanoled and cleaned all the time. So it does take quite a long time to get it through. We have about action, PCR, like 6 or 7 steps before I can get to the end result. And then it's just sort of a bunch of letters that don't mean a single thing until we do a bunch of computer work as well. So yeah, it does take a while. For the samples on here, maybe like months or...
Hold on a second, Georgia. Your microphone is just cut out, so I'm going to ask you to do that one more time.
Yeah, so as Georgia was saying, it's about 2 months, but it's a lot of hard work and a lot of time that put in, and I think they produce some fantastic results.
Yeah.
Great. So there's a follow-on question to this, and maybe it's the same question, and I'm not a DNA specialist, but the question was, how long does it take to sequence an average gene? And I'm going to give you 2 questions at once. And then the other question was, can you sequence RNA from your samples for proteins?
No, because some specimens, some of the mollusks, some of the work that Dr. Lisa...
So what we're doing with the samples that they collect is sequencing them to forward a reference library for my stuff. So David knows a bit about that. Yeah,
so some of the other stuff will get preserved in RNA later. So a lot of the mollusks get preserved into that for RNA work. So it's dependent on where we want to go further with the sample itself.
Yeah.
And maybe with the sequencing part, if we use traditional methods of Sanger sequencing, we're only looking at one specific marker. That step can take, it's a lot cheaper, and it can take a couple of days. Different species need more markers, and then with the new age of high throughput sequencing, that takes a bit longer, but there's a lot more of we have a lot more data, kind of what Georgia deals with, and there's a lot more work involved, and I think we have a great lab that kind of supports us. We all support each other, which is fantastic.
Awesome. So there's a follow-on question to the fish question from Rebecca. Does the pressure change affect the integrity of the sample, though? So do you see changes in the samples when they come up from the depth, or that really big pressure change sometimes?
Most definitely.
Yeah, Glenn brought up some fish. I don't know if you watched the live stream, Rebecca, was it? But there was a high-speed fish chase that was very rewarding. We got some beautiful specimens from that. They were looking quite good when they came up, but of course they lose their shape with the pressure and they go a bit...
A lot of the calcareous stuff, like I know you were talking about fish, Rebecca, but some of the sea stars or the hard corals, they maintain their shape because they've got that either got that exoskeleton or that internal. A lot of the crustaceans come up very well. However, some of the stuff like the sea cucumbers that are very soft, they will lose shape really quick. That's why it's so integral that we go from ROV, we put it straight on ice, we put it into the fridge, and then we slowly select which specimens that we'll process first. Of course, we'll usually go for the sea cucumbers first. Glenn will get onto the fish straight away, and that's because we're trying to photograph them still in the best possible manner. So most definitely different samples.
Yeah, but that's the awesome thing about having the ROV footage as well. So we can compare them to how they looked at the bottom at 4,000 meters to how they look when they come up on the surface.
Yeah, so traditional methods would have used dredging, just bringing stuff up, and we can really selectively sample with the help of the Smith Oceans Institute and the ROV team. It's just fantastic. Awesome. This is amazing.
Yeah.
The potential and the stuff that the Smith Ocean Institute does and helps us collect. It's really good.
Yeah, the ROV guys are fantastic. One thing I did forget to mention is the eDNA sampling. It gets quite rolly and rocky on the ship and quite easy. All my precious samples. Jason, one of the guys on board, built a nifty little over there. I mean, it's a piece of wood, but I didn't think of that. So that has saved me many litres of water and many nights of crying. So that was a good time. Thank you, Jason. Shout out.
Georgia, Jason has a question for you, actually, because he's sitting in the other room typing on YouTube. Is water from the ROV better than the CTD? Wait.
No, we don't know that yet, do we?
Don't know. I mean, it should be the same, really. The good thing about the ROV water is that once I sequence it, I can compare it directly to what you guys got. With the CTD, it's just a blind dump down into the ocean. It's not better or worse. If he's trying to get brownie points, then sure, it's better because he's controlling the ROV.
I think that's what he's trying to do. This is the ROV-MT war, right?
ROV over CTD. Don't tell any of the CTD people.
Georgia, can you explain? There's one more question I have from James, but first, can you explain what CTD stands for?
No. What does CTD stand for?
Anybody?
It's just an acronym to me.
Anybody? It stands for conductivity, temperature, and depth.
Well, there you go. I've just been saying CTD like a numpty.
That's all right. So for those of you at home, and I don't have a picture, sorry I didn't cue one up, but a CTD is more than just a bottle, 24 water bottles. It's actually an instrument that measures a lot of the properties of seawater. And so it's an instrument that we deploy over the side. It goes down through the water column and it collects dissolved oxygen, transmissometer, has beam attenuation, has a fluorometer, has all sorts of data in that instrument. And so we measure all of that as it goes down and comes back up, and on the way up we close these bottles at different depths for Georgia so that she can take these water samples.
Yeah, and the awesome thing about that is with the temperature data as well and the salinity and the dissolved oxygen, when I collect my samples, so I collect them at the bottom of the ocean floor. So say we did one, I think 2 days ago, and it was 4,000 meters. Then I collect some at 1,000, 500, 200, and on the surface. I can compare what I found in terms of species with the eDNA, but then I can look at all the data that the CTD manages to collect, so the temperature and stuff, and we can have a look and be like, oh, well, maybe that species was here because it prefers this temperature, this depth, this amount of light, all those sorts of things really come into hand. Figure out not only what species was there, but why it was occurring in those ecosystems.
Awesome. So I'm going to ask everybody at home, please send in your questions. We just have a few more minutes. I have one more question from James. I do not know what this means, so I'm sorry if I'm asking a blatant question out there. With CRSPR-CAS-9 developments, has your data seen more interest?
Is that CRISPR?
Nope. It's James Driscoll.
Oh, James is from, I met James
CRISPR.
at a conference.
Yes,
once.
CRSPR.
So CRISPR, We personally don't use CRISPR ourselves. I'm not sure where the museum is with that. So I can't really answer that myself.
I haven't used it.
No. We have used stuff like RadSeq technology as well, but yeah, it's kind of beyond me.
And me.
Okay.
A lot of these samples would undergo Sanger-seq, because a lot of markers have been developed already for a lot of these species, and some of the others would probably undergo high throughput sequencing to find new informative markers. But
that we can use.
I'm not too sure how to answer that, James, and yeah, it's nice to hear from you. I hope you're doing well in lockdown.
Okay, anybody else at home have any questions? I'm going to ask a couple while we're waiting, last couple minutes. Give me 1 second, bring up my list.
And I thought also mention, Janelle Ritchie couldn't be with us here tonight, so she's one of the fantastic technicians from the WA Museum.
Superstar.
And she's been really supportive and helping me a lot with trying to understand how things need to be preserved, subsampled, and what the role is and how they deal with a lot of the specimens. So it's really unfortunate that she couldn't be here with us tonight, but I have a lot of respect for her. So thanks, Janelle, for everything.
Awesome. Yes, we're sorry Janelle could not make it with us. So I have a question. What is the biggest difference that you find from working on this lab on the ship and your lab at home?
Well, for me, it's the rolling and the moving that I find the most different. The beauty of what I do personally with eDNA is that you can travel with those pumps. So those pumps have been a few places with me, and they're quite portable. They can run on battery power. So if I am out in the middle of nowhere and I need to filter a sample and we've just got a freezer, a bag or something, we can do that. It's quite portable. The biggest difference is on the ship, we are rolling and I do spill things quite often. So Jason has helped me and built this amazing contraption for me. The other thing that I have to be quite mindful of is on the ship, obviously, I can't just shove bleach down the sink. It goes out into the ocean. We have to be careful with how we store everything on the ship. We have waste containers for things. We're just really careful with the chemicals that we're putting into the systems. Deb and the MT guys have been fantastic in telling us how to do that and what to do properly and how to make sure everything's on board and legit. But that's really the biggest difference for me, is just trying not to spill. David's got a lot more differences, I'm sure.
For me, it's almost, the lab's fantastic. It's got everything that you need, if not more. We don't always have access to seawater in our labs.
I sometimes don't even have a sink.
So that's fantastic. But with the rolling, you really need to adapt to it. So obviously, if you're looking at specimens under a microscope, you need to time yourself. You need to take more constant breaks.
And with the pictures as well. We noticed the other night we were trying to take a picture of a specimen and roll, it would come out of the water, the photo was completely shot. We would roll the other way, it was perfect. So we were just in a nice little swing of things there.
A lot of patience, a lot of working together, and that's the other thing. Normally in our labs we kind of allocate certain times and the amount of people we'd have in a lab. And obviously on the RV Falkor, this lab is already very spacious, but with so many scientists working, really learn how to work as a team. And I think
just be respectful of each other's spaces.
That happened really quick. Everyone works together, works around each other, always puts in a helping hand.
So when I'm doing my stuff, I sort of clear everyone out of that side of the lab because we don't want any contamination in the eDNA samples. I'll bleach everything down. Everyone's super awesome about giving me that, and it's just been...
And also the same with our waste, so all the formalin will get put into containers as well, and we'll take that back. Same with any ethanol waste. Nothing goes down the sink. Everything's complied with MARPOL conventions. So any biological material, hazard waste, we all take that off the ship and that in the right method.
Awesome. So we have a question for you, David, from Rebecca. Have you collected any cool corals? And if so, what was the greatest depth?
So we got, there's different types of corals. So you have your soft-bodied corals, which are the octocorals. So we actually got an amazing, beautiful specimen today, if you go through the footage. So it was an orange one, so that was at 2,000 meters. We did also collect a cup coral, but I think that was only at about 1,800 meters. So that was a Desmophyllum I think a dianthus species. So that was an absolutely beautiful specimen. And if anyone knows me, they know how much I love corals.
Too much. It's disgusting.
So that was beautiful. We collected that on the first dive, but I think we've been getting some octocorals from 3,000 meters even. And that's, that is a range for some of the cup corals, the hard corals, but we just haven't had the right substrate yet to actually collect any of those. But yeah, it's just been beautiful. The colors are just amazing. I mean, to get these beautiful orange colors at 2,000 meters it's just spectacular. It's just beautiful to see, and to see the in situ photos through the 4K ROV camera, and then for
Stunning.
it to come up, it's just something that I don't think I'll ever forget. This experience, it's really, really cool.
Did you, David, have a close look at the black coral that you picked up from about 4,400 meters yesterday?
Yeah, we were that deep. Yeah, so we have been collecting. So black corals do have quite deep ranges.
Yeah.
Yes, and we have collected some beautiful black corals. And they're so delicate. And the ROV team does such a great job of collecting those, gently placing them in the ROV box. And just, it's just spectacular to see what state they come up in, and actually being able to take that through to the processing lab, to the wet lab, and being able to photograph it. It's amazing. It's absolutely amazing. So yeah, we've definitely seen a lot of black corals as well.
Awesome. So we have one question. This one's again from James. How are you both handling seasickness? They're going to find out tonight.
Yeah, come on now.
With all
Drugs, yeah. No, we do take seasickness tablets occasionally. For the first few days, I was taking one at night, and then that's kind of helped me. But we've been really blessed with weather up until now. We've been really, really lucky. We haven't had too many bad days. I think the next few days are going to be a little bit awful, but I think we've been okay, David.
I think Captain Pete's doing a great job with his team, and I think they're keeping us out of big swell. And no, it's been good.
And the food's just been too good to not want to eat.
Yeah, how can you be sick? How can you be sick when the chefs are cooking these fantastic meals? I mean, I don't think I'm looking forward to going home.
No, I don't want to.
That's great. Awesome, guys. Just for those at home, I put up a picture of the CTD. It's from our previous cruise. And you can see some people here sampling. So this is what Georgia and her team are doing every day. So there is a question for our ROV pilot. I just yelled at him in the other room. I'm not sure if he's going to come in and answer it. James, the ROV is faring great. Yes, we got very close to the max depth rating of the ROV over the last couple of dives. We were under 4,500 meters, though. So here, Jason has just stepped into the control room with me. I'm going to hand over it, but our family at home would like to know how the ROV's faring, having gone very, very close to its max diving depth.
Hey, James. Going down to 4,500, which is the Sebastian's limit, is always a scary time for us. We can go many months and almost years of never even getting close to that, and the sub will work fantastic. And once you start going down and getting those little bit extra squeeze, it'll start exposing all the weak points that you have. And we just found that out the other day. You might have noticed during the dive, we started having issues with our cameras, and one of our bulkheads, fiber optic bulkheads, had actually cracked. One of the really small microspheres had gotten shattered, and we couldn't use the 4K or the 1080p camera anymore. It ended up being a quick fix, but when you're going down to 4,500, we didn't have enough time to be able to make it up and go back down in the same day. But that's why we were able to come back pretty easily the next day.
Thanks, Jason. Okay, so any last questions from home? While I'm looking for the last minute questions, I have a question for both of you. If you could bring one thing from home and put it in that wet lab, if you were designing your favorite, most amazing wet lab on a futuristic research vessel, what would you want?
I want a cat run so I could bring my cats. No. I think, the most futuristic, most amazing lab for me would obviously be a DNA lab, unfortunately. It would just, all of this ecology stuff out, all the biology out, and then just PCR machines, a sequencer, and it would just be clean. Everything would be nice and clean.
So Georgia wants a DNA room.
I want a DNA room. Selfish. David?
I think that's a great idea. But I also have a wish list on top of the DNA lab. If we could add an SEM, so a scanning electron microscope, oh, that would be fantastic.
Yep. Just get rid of those engines. Put one of those right in the wet lab.
How many engines do we honestly need? Like, let's just make it a sail ship.
Awesome. Great, guys. Thank you so much, everybody, for joining us. I have one final question from home. My father is asking you, what is the most unique sample you think you've collected so far?
For me, it's all water, isn't it? But at the end of the day, Casey and I, the girl who's a master's student who's helping me,
Georgia, I think you should introduce Casey.
Casey, would you like to turn the camera around on yourself?
Casey's been our camera woman almost every single night. So thanks to Casey. Casey is on her second expedition with us. She's a trooper.
Casey gets up every morning with me at 3:30, 4, no matter what. She's there helping me out. I'd be in serious trouble without Casey. We were talking the other day about how cold the water was, and we just realized that it's come from 4,500 meters. Like, that's just incredible. It's so awesome. The other highlight for me has been, sorry everyone else, but Glenn brought up these fish and I lost my mind. Like, I thought they were the coolest things on earth. I've never seen, like, from a nightmare. They were so weird looking. They were those big, long, white ones. I don't know if you remember what they were. They were itchy and weird and horrible, but fish. Beautiful. Yeah, blobby little weird things, but they were a highlight for me.
That's such a hard question, seeing all the specimens come up. I guess I won't create bias by picking corals, but I think I'll stick to the Cnidaria family and probably the sea lily. So it was just seeing it in situ. It still came up pretty well intact.
Yeah.
And I think that was just amazing to see its big globular stalk, gelatinous stalk, and just that was just amazing.
But even some of the tiny little worms and the crustaceans that everyone brings up,
yes,
like
the polychaetes, the scale
it's
worms.
It's incredible. Like, I've never seen anything like this before.
I have to say, some of the stuff that Greg Rouse was working on, the scale worms, like their colours and their plates, it's just fascinating. It's something that I haven't had experience with, and but yeah, he showed me a lot of things, and it was really, really cool. They're beautiful creatures. Everything's beautiful. Awesome. From the tiny mollusks, the little crustaceans, to the big stuff.
Big ugly fish.
Yeah.
All right.
Hard question.
Yeah. I want to thank both of you for joining us tonight. I'd like to thank everybody at home for joining us. Remember that you can always watch our ROV dives live on YouTube and Facebook. You can find out more about this expedition at schmidtocean.org. Tomorrow, I'm not sure yet if we are ROV diving. That's a little late breaking news. We are currently heave 2. We were mapping until about 15 minutes ago when I got notified from the bridge that the weather is not even good enough to map, so that's usually our last resort. So we've got a bit of weather for the next few days. When we are back online, please join us. We are also here Monday night, again, the same night for us, maybe morning for you, maybe afternoon for you. Our next one is going to be a live tour of Falkor, so we will take you on an exploration of the ship. Some of it will be pre-recorded because it will be dark on the bridge, but we will show you that. I think we might actually get to talk to Captain Pete. I hear his kids have been requesting a cameo, and hopefully some other crew members will join us and explain to you a little bit about life at sea, their work here on Falkor and what they do, and we can show you a little bit more about our ship. So thank you very much to David and Georgia.
Thank you. Thanks, Deb. Hi, Mum and Dad.
Stay safe, everyone.
Yes, please stay safe, everyone. Try to stay in. Wash your hands. There's an amazing message from ROV Sebastian that our pilots have put together for you. Wash your hands, and please stay safe and healthy. We are doing the same on Falkor. We have been out at sea now for, I've lost track of the number of days. So we have
3 weeks now?
well quarantined ourselves. Nobody is sick on board. And we are continuing the expedition to be able to bring you these images from Falkor and to allow scientists to continue with their work. And we will keep doing that as long as we can. So thanks for joining us, and we will see you again Monday night.